With modern sun, you can see its high oxygen production, which is expected, followed by a dark period. Proxima Centauri is about M5.5, so a late M dwarf, where we didn't see any oxygen production. But in the early M dwarfs, we got our target. Like we can select those in terms of doing further UV experiments. And interestingly, when I did the UV experiments, I did bunch of, but I have shown one slide where I did a series, kind of a sequential UV treatment. So again, on y-axis we have oxygen production rate. On x-axis we have different treatments. The green is just the light treatment, light only control, where I expose the lichens to 42 hours of just the visible IR flux, not the UV, followed by a background UV. So these M dwarfs have low UV during the quiescent period, but they, as I mentioned earlier, that during the flares the UV frequency increases. And the flares, like if flare is more intense, it's more rare. The flare that is less intense is more frequent. So just to simulate a more kind of extreme scenario, I expose them to 42 hours of flare event. And on top, we have the percentages like percent change from the control. And you can see with the flares, the oxygen production rate was significantly suppressed. However, following the again background UV exposure, they were able to recover back, which shows the flares will suppress the productivity, but eventually when the flare is over, they may be able to bounce back. Now we showed that it's not just survival; they can be active as well. But can we think about now the oxygenation of a planetary biosphere? So I have assumed a lot of things to do this, but just briefly, I have assumed like we have a Earth land to ocean ratio. We have about 70% maybe ocean, 30% land, and then because they will be light limited, I have assumed three different conditions that lichens maybe life is on the substellar point, and then on the mid hemisphere, and then uniformly distributed over the planetary biosphere. And this framework was derived from Lemur et al.'s paper 2018, and I used Earth-based sinks and Earth-based organic carbon barrier rate, which may be a underrepresentation because lichens does not be as much. They don't have a complex root system to maybe have more carbon barrier rate, but this is just an assumption to show you where I'm going with this. So under just a substellar scenario, on top we have algal lichen and below we have a cyanolichen. And on the y-axis I have active fraction of lichen, and on the x-axis we have the total land coverage. So per hemisphere we will assume like 14% of land is covered by the lichen. So 100% on the x-axis means the whole land is covered by lichen, and 100% on the y-axis means that they are always active, which is not ecologically true, but just to show that they are not light, nutrient, or even water limited. So again, if you see the green solid line here, that is the control, and above that line means they are oxygenated, like they have crossed the threshold of O2 sinks to produce net oxygenation. And below that line, the net threshold is below, so they can't be oxygenating the biosphere or planet if we think about. So what you can see is like even if we assume a more realistic scenario, maybe the 50%, even that's a lot, 50% land is covered by lichens, and you assume, let me see if I can point, assume 50% the activity, so it should be somewhere here. So what it shows is that just barely under light and even quiescent UV, they were able to cross the threshold. Under all the conditions, the threshold kind of shift. We needed more activity or we needed more land coverage. Even in case of cyanolichen, the curve is shifted a bit because they were more productive under our conditions, but still the same scenario, 50% should be somewhere here. And the same thing with the mid hemisphere because it's even less productive. And the worst case happen when you think about the uniform distribution, like they're distributed kind of uniformly, which may be ecologically, like because we have just assumed that the more relevant that they are distributed that due to weathering near the ocean, if there are small islands, they can be having more nutrient kind of exposure. But what it shows is that in this condition, they were not able to cross the threshold. And with that, I just want to conclude that maybe when we think about here, we are thinking about flares, they may not eliminate the photosynthetic life, but they may result in net non-oxygenated biosphere. And if you think about now false positives as well, that we have abiotic oxygen production, so it is difficult to separate like where the oxygen source is coming from. That thing that we need to incorporate multiple evidences, which is why I think surface biosignature project is one thing, and maybe albedo or the multiple evidence we need to rely on. So not just think about UV, but we need to think about all the stressors that happen, that we can even speculate from Earth, that we know the UV flare activity. We also, someone talked about magnetic field yesterday, but if we have to assume that if there is no magnetic field, there could be radiation as well on these surfaces. To just to do use this stressor to maybe if we can understand something about life as we know it. So with that, I would like to thank you everyone for listening and my collaborators at NASA Ames and GSFC and Blue Marble Space. And I'm happy to answer any questions if I have time.
Fantastic timing. We've got 2 minutes for questions. Anyone?
Really cool experiments and great talk. I just as an astronomer, do you have a theory as to why the lichens shut down under flare activity? What's happening to them? Are you actually able to kill them with your setup, or what's going on?
That's very kind of interesting question. So the one thought I have is because, like maybe this diagram also help. So when the UV interact with these organisms, right, they have these sunscreen compounds. So what happening is likely is that UV is blocked, which my earlier experiment showed, but they form a lot of reactive oxygen species because even if there is no oxygen in the setup, they are still producing oxygen through the photosynthesis. So maybe those things are damaging either the proteins like photostatic pigments. So that's the only reason, like, and they need some time to recover that back. So maybe, and also there's another thing that I didn't discuss. These are much more resistant in desiccated conditions. I exposed them to even 60 watts, which is 15 times more than Mars for 3 months, and they were fine. So it also depends on what state the lichens are under the condition. Like maybe if this extreme superflare event and they are desiccated, maybe they will survive more. But it's more of like the regular cycling between the repairing the proteins versus the damage.
One more question.
I thought that was a really great talk. I had a quick question about your physiological experiments. The error bars seemed pretty wide. And I know
Yes.
I do similar things. I know it's super tough. Do you know what you could attribute that
Yes.
variability to?
Definitely, because these are natural specimens and we don't even know, like some are maybe young, some are older. So older one will be like more susceptible, I guess. So it's more of because the natural variation exists. Even in my controls, you can see the variation and it's got selected more as you go on the experiment. Yeah, thank you so much.
Thanks, Tashinda. Thank you. Great. Next up, we have Ligia Coelho with a 10-minute talk.
Okay. I didn't know there was a thing like this.
Can you hear me well? Oh, yeah. So is it manual? I'll just go manual with this. Or... okay, yeah. Okay, it doesn't work well. Okay. Okay. Good morning on this beautiful Sunday. I know that everybody's already very drained, even though Absacon is about to start, but let's try to brace ourselves and continue. So I'm going to talk about surface and atmospheric biosignatures in the era of ELT and HWO. My name is Ligia Coelho. I'm a 51 PhD fellow at Cornell, starting in Caltech in 2 weeks. And I want to just represent the paradigms of live detection so that we put these things assembled very well in our brains. So we've talked about atmospheric gases. This is where we are right now, in the sense that we have the technology. I don't know if this is going to work. Oh, it will. In the sense that we have the technology currently to try to detect them. We haven't yet detected atmospheric biosignatures, but James Webb is here, and we are very happy to have it and to work with it. The problem is that transiting planets are not the most frequent, so they are very rare. And so the direction of the field is going towards direct imaging, reflection spectroscopy through direct imaging, where you're not too limited by where the position of the planet in relation to us and the geometry. You can just take a picture of it, right? And so in light of that, there's telescopes that are being designed And for instance, the Extremely Large Telescope from Europe is going to have first light in 5 years. And I put it in comparison in terms of size with the La Sagrada Familia, because I like to think one of them will finish first. I just don't know which one. But adding to that, HWO, we've talked extensively about here, is also going to come and revolutionize biosignature detection, but we also have life being designed right now. And the idea is that all of them are covering something different, and they are going to be highly complementary. The Extremely Large Telescope is going to bring the high-resolution spectroscopy angle, which allows us to see things that low resolution doesn't, or space-based resolution doesn't. But then HWO and light are going to cover different regions of the spectrum. So we are all covered, right? More or less. So this opens the opportunity for us to look at surface biomass, which is fantastic, and this is why we are here. And this is where the upcoming telescopes can take us. But the problem is, astronomers are dependent on retrievals being reliable. The problem is interpretation of these biosignatures. Just one single biosignature are always going to be a big headache. It's just for a retrieval that Anna Grace talked beautifully about and showed data about is an inference of planetary properties given a model or an observed spectrum. But a retrieval is only as powerful as the diversity of spectra it knows, and only as limited as the ones it lacks. And if you try to cherry pick which ones you add to your model, you're going to get in big trouble. And I'm not going to extend more on that. Adding to this, though, this was a big source of inspiration for me. With a background in microbiology, but with now 5 years in postdoctoral time in astronomy, this was a problem that really called to me. How could we expand on this knowledge of biodiversity in astronomy and make these models more reliable? So my method, I covered the Earth in fieldwork, measuring the albedo, the surface reflectance of these locations that have biology. I mean, every location has biology, but you know what I mean. These locations that you have visible biology that could be detectable. I also get some of these samples back to my lab, and I do measurements. I also grow them. And with all these measurements, I build models that are sensitive to resolution to all these different instruments and these different telescopes. But adding to these reflectance is not the only thing that we add to these models. We add also the context, because the fact that we go to the field and take all these measurements, we also take a lot of contextual environmental data that we can input to these models. So one of the papers that came out of this work was Purple is the New Green, which was the first spectral database of an oxygenic phototrophic diversity. To be clear, this kind of spectra already existed before. What we wanted with this paper was to bring diversity and show the diversity of the features that this very diverse group of bacteria can have. And this also added with the models database of purple planets that we can now think about for instance, orbiting around them stars. And the samples were in great quantity from the Sipuisit marsh. And so the other novelty of this paper is that we started working with communities instead of pure cultures. And this allowed me to start developing my model that is resolution is still being developed, but I've been playing with it and adding to my papers already, where I can show the difference on how these different instruments with different resolutions can actually target these biosignatures. This paper, because of the title, got a lot of press, but what I want to call the attention to, just for a comedic relief, is that there's a lot of nice press, but then this one freaked the hell out of me because I'm way too young to understand what purple people eaters meant, and I really genuinely thought they misunderstood my paper, and I was going to get in trouble, and I'm on the J1 visa. I definitely didn't want that. Anyway, but now I understand this was actually a nice cultural reference. Adding to the M-star systems and the purple is the new green, I built other spectral libraries that I'm not going to talk about today, but they are all available. Astronomers can download them and hopefully use them in their models and retrievals. But it's not only about the spectral library. We also learn a lot about just looking at the spectra. We learned, for instance, that in comparison with false positives, like, for instance, minerals, biopigments have much more features that can be probed for. So each peak that you see is an opportunity to detect life, and not all of them are masked by the atmosphere, as opposed to a mineral, for instance. But the other thing, and maybe the most important thing we learned, is how dryness enhances the biosignature intensity by over 60%. And this means that if we have measurements of both hydrated and dry biopigments, we can allow astronomers to be more reliable on their models because they can add the hydrated to the oceans and the lakes, and they can add the dry to the deserts like Terrinder's beautiful work, or to the icy surfaces. But then we also learned that when retrievals compared modern Earths with these microbial icy and desert worlds, they had a great preference for them because of the intensity of the biosignature. And this is important to inform maybe the future targets. Maybe to start probing the surfaces, we should start looking maybe at the easier targets where the intensity will be higher. I just want to finish with one idea for our white paper. So I've talked about atmospheric gases, surface biomass, but there's also atmospheric biomass. There is another paradigm of life detection. This is life that lives in the atmosphere and in the clouds. In our own planet, we cannot see it because of the low cell density, but that could be an artifact of Earth. There's other papers that claim that with different Earth circulation, you could have a more agglomeration of these cells that could be detected. Because of that, we built the first spectral library of these kinds of organisms directly isolated from the atmosphere. But what I want to bring attention here is that they could be photosynthetic too. So we are not claiming there's photosynthesis in the atmosphere. That's a big question if there's activity in the atmosphere. But there's clearly capacity for isolate creation and production of biopigment, which is what we need. And so I think that for this white paper not to get old too quickly, considering these projects are now being studied, big papers are coming out of these groups that are studying these organisms in the atmosphere, I think we should mention at least the possibility of these organisms in the atmosphere also being a biosignature. To finalize, I don't think the paradigms of life detection is what is the really important thing here, is actually learning about them as alternative lines of evidence for life. Each of them individually are going to not be too robust of a detection for a lot of reasons, and I'm going to put my dog in that fight if you want to talk about it. But I think we can use them as alternative lines of evidence for a detection. And so this is my last slide. In line of that, I want just to mention that I started my research group recently on biosignature detection and interpretation, and we are focusing on alternative lines of evidence. This is my already graduated students. Amazing Honan Gong that is starting as a PhD student in ASU, Neil Jerome that is starting as a master's student in Madison, and also my current students, my other ones. So thank you very much.
Thanks very much, Ligia. We've got time for one very quick question while we change slides.
Thanks, that was great. I may have missed something really important. The purples that you're envisioning for the Archean, are they purple sulfur bacteria principally?
So I didn't make the paper for the Archean, it was for M-stars.
Okay.
And they were purple sulfur and purple non-sulfur.
Okay, so we can talk a lot about that. There's an issue of where that plate is, and Nikki and I talk about this all the time and others, but more the case is if you chose the Archean, there is almost no sulfide in that water column because there's no sulfate.
Mm-hmm.
To get to sulfate reduction, you have to have really high productivity. So it would be nearly impossible to turn an ocean purple through sulfur bacteria.
I haven't thought much about it because I was not to the Archean. Again, I was doing a modern planet but around an M-star.
It would be true in the Proterozoic as well. To get to sulfate reduction in a redox ladder, you need to go through a lot of oxidants first, and then you have to have a lot of organic matter.
But if you have a lot of years of evolution down to a modern Earth stage around an M-star, do you think you would have the same problem?
You just need sulfate, and you need organic matter. You need an electron donor. You need a microbial ecology, which is hard to do across a broad portion of an ocean for sulfur.
Mm-hmm. Yeah, it's a good thing. We can definitely discuss that.
Sure.
Yeah. Thank you. And I'm sorry about the time.
Don't worry, thanks again, Ligia. Okay, next up, another 10-minute talk from Elizabeth Alistro. Take it away.
Thank you. Okay. So I'm Elisabetta, and today I will show you some of the work that we've been doing in Padova about oxygenic phototrophs exposed to simulated planetary conditions. So we are all here because we want to understand whether oxygen photosynthesis could be possible elsewhere and whether it could give us the biosignature that we know on Earth, right? So oxygen in the atmosphere and vegetation like that. So to address the possibility of this metabolism to function elsewhere, we decided to address the worst case scenario for photosynthesis likewise, which are M-dwarf stars. So M-dwarf stars are much fainter than other stars, and they emit very poor visible light and mainly emit far-red light, contrary to the sun, which is a this type of star which emit a lot of visible light. And indeed, on Earth, photosynthesis evolved a series of pigments that specifically harvest these wavelengths up to 700 nanometers, while far-red light, being less energetic, is poorly absorbed by oxygen phototrophs in order to drive this metabolism. But if you look at the spectrum of an M-type star, you can find niches on Earth that receive a kind of irradiation that is similar to this one. And in these niches, we often find organisms that perform oxygen photosynthesis. So somehow they are coping with this irradiation pattern. And this opens to the possibility that this metabolism can function also around M-type stars. And to address this, we have developed in our lab a setup that can simulate starlights. We can reproduce different starlights. And here you have the M7 spectrum that we are reproducing, coupled with a solar light simulator and a far-red light simulator. And the starlight simulator is equipped also with an atmosphere simulator, which is basically a small growth chamber that we can place underneath the different light sources, where we can inflate a desired atmosphere. And inside the chamber, we have a sensor for CO2 and O2 in order to monitor the oxygen evolution and carbon dioxide consumption in real time when they are exposed to the star spectrum. So during the last years, we've been testing many different organisms, and today I will just go into details with cyanobacteria, because they evolved oxygen photosynthesis on Earth, and they can do a lot of interesting things with their light harvesting systems in terms of acclimation to different kind of spectra. So we tested the cyanobacteria with these three spectra, so solar, M7, and far-red light. And you can see that the amount of visible and far-red light in the three spectra is highly diverse. And the atmosphere that we are testing is an anoxic atmosphere enriched in CO2. So we have like 5-8% of CO2, and the rest is just nitrogen. So we tested 4 different cyanobacteria that can do different kind of acclimation. And I will go through the physiological responses that they give, and then I will address the biosignature that they could potentially generate. So the first one is WALZ 6912, which is a far-lip strain. So under far-red light, the strain can synthesize red-shifted pigments like chlorophyll D and F, which allow them to grow just relying on these low energetic wavelengths. And this was the case when we exposed them to the M-dwarf starlight. You can see from the in vivo absorption that they synthesize, it's not this one. They synthesize chlorophyll F and D, and they are using the high amount of far-red wavelengths that they have available in the star spectrum. Then we move to a strain that cannot do this, so it's a visible light user. It cannot produce red-shifted pigments, and in the M-dwarf star, we don't see any major acclimation. So this organism can just rely on the visible light that is available, which is very, very low. The third one is probably my favorite, because it can perform both far-lip, so it synthesizes chlorophyll F and D in far-red light, but it can also perform chromatic acclimation type 3, CA3. That is the one that you have on the bottom right. So basically, it interests the phycobilisomes, which are the light harvesting systems. So in green light, we have a lot of phycoerythrin in order to maximize the harvesting of green light, and in red light, they produce a lot of phycocyanin in order to maximize the harvesting of red light. And this what was happening under the M-dwarf star. So these organisms were not taking advantage of the far-red light that is available, which is much more than the red light, which are they actually using. You can see that they are increasing the content of phycocyanin and just relying on the low amount of red light that they have available rather than harvesting far-red light. And the last one that we tested is Agouron marina moss beach, which is one of those strains that has a high constitutive level of chlorophyll D in their photosystems. And so it constitutively harvests far red light. And you can see that in neither of the spectra that we tested, we see significant acclimation. This is, let's say, already suited for this kind of spectrum, and it constitutively harvests far red. So this strain gives us very different kind of acclimation responses. They do a lot of fun stuff. But when it comes to the impact that they could have on the anoxic atmosphere, we saw that they are all producing sensible amounts of oxygen in the M-star spectrum, irrespectively of whether they can or cannot use far red light. And I think this underlines the importance of simulating like the true parameters, because if we are just relying on whether they can or cannot harvest far red light, we are missing a big part of the picture. Because organisms that cannot harvest far red light are producing oxygen. Organisms that can use far red light are producing more oxygen in the amber star, which is extremely exacerbated in Agrisera, which produces more oxygen even than in the solar light. And organisms that could use far red light rather decide to use just red light instead in order to evolve oxygen. Then coming to the surface biosignature, we asked ourselves whether the red is a must. Okay, so here you have the reflectance spectra of 6803, which is the one that can only use visible light. So it's a, let's say, regular red edge. But when it comes to organisms that can acclimate or can, let's say, harvest far red light, you can see that Chlorophyllopsis has this bump here that is due to chlorophyll F and D. And when it comes to Hydrochloris marina, the red edge is shifted toward longer wavelengths. So different kind of acclimation and adaptation strategies can give very, very different spectral signatures. So now what we are doing next is we are coupling this simulation with UV flare simulation. So we developed this UV flare simulating lamp that can reproduce faithfully the UV flares coming from different kind of stars. And we are doing preliminary experiment, so integrating this with the simulation of the star and the anoxic atmosphere. So just to conclude, what I want to stress today is that photosynthesis on Earth has evolved mechanisms that allow to thrive in niches that are considered unfavorable, likewise, and that under the M-star spectra, we have oxygen evolution irrespectively of the acclimation strategies, which rather have an impact on the reflectance signature. And last but not least, we really need to test these parameters in order to understand the true limits of photosynthesis. Thank you.
Lovely talk. We've got a couple of minutes for questions.
Thanks, great talk. So yeah, it's nice to see these sort of far red light acclimation strategies in comparison to the regular chlorophyll A type systems. I mean, at the end of the day, you're only talking about something like a 50 nanometer shift in that red edge. Do you really think that's like substantial in the context of looking at, you know, the flux of M-dwarf stars?
Do you mean if we could really expect a shift in the spectral signature to be detectable?
Yeah, I suppose, or even if it's even like a substantial enough change where, yeah, I guess detectability, like above the noise would be.
Yeah, I think it is something that we need to take into account when looking for reflectance biosignature in order to avoid like saying this is a negative. It's something that on Earth is possible. So if we see something like that, we cannot say it's not oxygen photosynthesis. Mm-hmm. Okay, and this is also important to compare with the simulation on the experiment on false positive. So we need to integrate these two knowledges, let's say.
Yeah, it's more complex than what I'm. Yeah.
Great. Final question over here.
Hey, great talk. So I was wondering, like, when you were thinking of UV flares, because these organisms doesn't have like much natural sunscreens, are you thinking of using iron as a shield, or like what are your plans for the shielding?
We are using quartz glass in order to have all of the UV passing through, And we are using actually in the project where we developed the lamp, we plan to use hydrothermal and desertic cyanobacteria, which have more sunscreen strategy than most cyanobacteria. So hopefully they will be fine. I think they will be fine. Thank you.
Thanks again, Elizabeth. Okay, final talk in this session is a 15-minute keynote from Carl Stapelfeld.
Testing 1, 2, 3. Can you folks hear me? I think you can. So good morning. Thank you to the organizers for inviting me. The Exoplanet Exploration Program at NASA Astrophysics has always been adjacent to astrobiology. We'd like to make it more integrated. So that's part of the reason why I'm here. I think this is the only programmatic talk at this meeting. And by programmatic, I'm going to be talking about how we integrate together different interests and facilities to get a overall ability to advance exoplanet science and therefore astrobiology. I've had the good fortune to be part of the DARES task force, leading focus area 3, which is biospheres, worlds and planetary systems, the coevolution of those. So Nancy asked me to give you an update on DARES about where it stands right now. All right, so DARES is the next astrobiology strategy document that NASA headquarters is working on with a large group of community members. It's meant to replace the 2015 internal planning document that was put together by Mary Wojtek. It's a non-consensus strategy and it's intended to integrate together a lot of other planning documents that have been produced by the community over the past 5 or 10 years. So the reason to do the DARES effort now is that the different strategy documents are all kind of coming from different directions. We have the astrophysics one from 2021, the planetary astrobiology one from 2022, heliophysics. We've got Mars exploration strategy reports that have come out, and there's lots of issues for the NASA workforce we want to work on internally. And there's all these results from the missions shown in gold here since 2015 that are affecting the state of the field, many of them extremely exciting. And these upcoming opportunities with these large missions and observatories, and perhaps a big opportunity upcoming with the return of humans to the moon. Not that we're going to find life there, but maybe evidence of volatiles or maybe fragments of the Earth or other planets that have landed there. So how can we best build on all of these inputs and advantages of the new opportunities? So there's a whole mob of people that are involved in this. This is the leadership face page. Including myself in the lower left and the headquarters leadership. And the overall group is listed here with 3 leads at headquarters. We've got 2 co-chairs from the community, 11 ex officio NAPS representatives. That group is going to be taking the report written by the group on the right, and they'll be implementing it internally with NASA to decide what to do with it. And then a total of 63 people, most of whom are from the community, are involved with this. Here are the 9 focus areas that were identified, and these all mean that there are subcommittees working on these topics with a lead, executive secretary, ex officio NASA person, and 3 or 4 community members each. So I'm on number 3. Some of these are related to research, shown in blue. Some of them are about the state of the field in green, and some of them are more on missions and technology. So the activities and the schedule, this all started in the end of 2024 with community white paper call with 130 submissions made. The first task force was assembled from self-nominations from the community. A workshop was held in May of 25, and those white papers were all synthesized into a smaller set of documents to handle. I'm on task force 2 that was assembled last fall for nominations, and now we are in the process of presenting our preliminary findings at AppSciCon on the Monday evening sessions. I hope you'll come to that. We want to finish writing the full report by September and presenting it to the community at the AGU meeting. So now I'm going to jump from the DARES activity to something that we do in the exoplanet program, I like it to be a template for what DARES does and maybe what this group does. And this is the idea of tracking science gaps, areas where if the community worked in these areas, it would advance the future NASA missions and current NASA missions, the scientific return from them. And so this is meant to cover the whole area of theory, lab measurements, simulations, and observations. NASA headquarters uses our exoplanet science gap list for the XRP proposers, giving the proposers a chance to relate what they want to do to the goals that NASA has. We are not smart enough to think of everything that would be good in exoplanet science, so you should always propose your own idea, and because you'll maybe come up with something that we didn't. But we want to help the selection officials, review panels, and the proposers understand what NASA would like to see. We update this annually. This is the 2026 report, which is on archive here, and we'll be doing this again for 2027. So what do the science gaps look like? Contents for the 2026 document. Notice gap number 16 in the arrow is the biosignature gap. That has been looked at by some astrobiologists. Nancy was good enough to give us some comments on it, but we'd like to have more astrobiologists look at that. So if you have a chance to look at our document, please see that. And these are all grouped by what is the capability that we want to have, or the measurements we want to make. What is the state of the field today with respect to that? And we want to list in our gaps what we know of that's happening now, so that a proposer not only knows what the goals are, but maybe knows what research to check on so they can build from it. And what we don't list as a mitigation in progress is what the community should propose. So this again is a 60-page document. And so there are gaps that pertain to HWO. I don't want to dwell on the first two, they've been around for a while. The third one is about what is the mass precision that we need to measure planets in the habitable zone in order to support this interpretation of spectra. So we're actively working on it in the exoplanet program. We had a workshop on that in April. We'll be doing another one in the fall. We want to know what the mass precision goal needs to be for either astrometry or radial velocity. And then one of the things I want to make sure that this community knows about is that the wavelength coverage that HWO is going to be able to get is going to be different for every star. And that is because the inner working angle of the coronagraph, how close in you can look, is a function of wavelength. It grows larger with wavelength. So you have widely separated habitable zone, you can cover all the wavelengths. If your habitable zone is sort of close in next to the limit of the coronagraph, then you'll be able to see it in blue wavelengths, but you will not be able to see it in red wavelengths. And so this effect is shown on this chart here, where I'm plotting for the total number of 164 stars, what wavelength HWO, the nominal coronagraph design, would be able to go to. And you can see that across the visible out to maybe 0.9 microns, you can do all of the targets. But then this is for an Earth analog at the inner edge of the habitable zone. The inner working angle starts to cover up your planet, and so you lose the ability to see it. And so by the time you get to 1.6 microns, you're down to only a quarter of the sources. So this is something that you should take into account in your modeling. And when you say that HWO could do 25 habitable zone rocky planets, yes, but not all to the same red wavelength cutoff. And so this is similar for the other kind of metrics that I think we talked about, like the ability to do ocean glint or the ability to do the rotational modulation of the planet to see surface features. Those are not going to be doable for every target either. So we have to consider every star and its habitable zone planet individually. So the last thing I wanted to get to was the discussion here of the targets for HWO. The mission is not designed yet. There's no final target list. A 2040 science team will decide that. But NASA headquarters gave the community an opportunity to do precursor science for HWO, and lots of folks wanted to work on targets. So Eric Mamajek, my deputy, and myself put together this list, selecting according to 3 non-scientific criteria. First, the planet in the habitable zone has to be bright enough to do spectroscopy in a reasonable amount of observing time. That means 31st magnitude or brighter. That's like a galaxy in the Hubble Deep Field, if you know that reference. So be bright enough to do spectroscopy in a reasonable amount of time. The second thing is being able to not be too difficult, a brightness difference between the planet and the star. We've been aiming for 10 to the minus 10 contrast in our laboratory development of coronagraphy. And so you can go below that, but you can't go a lot below that. So don't ask for a planet that's too hard in terms of a contrast. And then you want to make sure that you get enough planets that the goal of the Cato survey set of detecting 25 would be achieved. And so that means you take the widest separation habitable zones and keep adding them until you've gotten to about 100 or maybe 150. And then you say you're fine with the list. Now, we'd love to have more targets than this, but it will require a bigger or more aggressive HWO that would cost more money. So this is like a minimum target list. If HWO is made more capable, then you could go beyond it. And so what I want you to see here is on the bottom panel that the stars that you can do are, when you apply these criteria, are really limited. There's very few M stars. Essentially, only the brightest 3 M stars in the sky are accessible here. And then there are fainter ones that you could see, or ones that are closer in, you could see maybe in the blue, but not all the way out through the red optical. And so you can see it's dominated by F and G stars, although tier C is full of F stars where the contrast is a little more difficult. So this list has proven to be quite popular, that there's like 65 citations to this document in the community in the past 2.5 years. It's like the most cited paper with the term Habitable Worlds Observatory in the abstract. And so I wanted you to be aware of this because the exact targets, for at least the best ones in tier A, are almost certain to be observed. And the community will observe these targets with ground-based telescopes, with space telescopes and X-rays and so forth. And they'll scrutinize this list and find out if there's some that should be thrown out for one reason or another. And so this list includes a lot of data and tables available at the NASA Exoplanet Archive. So the location of the star, various names, what the separation in AU and in milliarc seconds is for the inner edge of the habitable zone, the contrast and the apparent brightness of the Earth analog and reflected light. It has the motions, that the amplitudes of the astrometry or RV signals that the Earth-like planet in the habitable zone would produce. And we track whether or not there's any known disk or infrared excess dust in the system, which can produce a background exozodiacal light that makes the spectrum more noisy. So all that information is in here, and I think that's the kind of thing people are using. This has been used for ocean glint papers, it's been used for RV papers. A lot of folks are looking at it. And then we've thought that it was important to get the public aware of what these likely HWO targets are, because we're asking folks to spend a lot of money to Congress to support the project. A lot of these are naked eye stars that you could go out in your backyard with your scout troop and try to point people to. So we've got a public-facing version of this HWO catalog as well. We want to show people where we're going to explore. And so you can go here to the NASA science website, and when you go there, you'll be able to see what all the targets are, their brightness, what constellation there is, whether any planets or disks are known in the system already. And so this is like one of the targets here, Beta Hydrae. So it gives information on the star that's in a public-friendly way. And we've had a couple of folks work on what is known culturally about that star. Many stars on the list appear in science fiction novels and so forth. And so we've accumulated that information. Many of the stars are anonymous with just not even a name, just a catalog number, and no literature discussion of them. So you'll find a whole mixture of different types. So what I wanted to end on here is suggestions to photosynthetists. Is that what you want to call yourself? I thought I shouldn't call you photosynthetics, so photosynthesists. In terms of getting your inputs into strategy of the various kinds that's happening, try to get gaps or the whole workshop white paper, done in the next couple of months, because NASA DARE's strategy for writing its report is by the end of the summer. So to get noticed and mentioned in that would be a nice schedule to keep. If you're coming to AbSciCon, then please come to the Monday evening session at 7 p.m. It'll run for 90 minutes. DARE's will be about half an hour of that, along with an overall discussion of the state of NASA astrobiology. And so at some point, the HWO community science and instrument team is going to be deciding on their draft requirements. Vicky's here as the representative of that. And so if you have inputs on that, like in particular, is it okay if only 25% of your habitable zone planets are visible at 1.6 microns? If that bothers you, maybe the CSIC should know that, and there should be some other number, like half the targets. How many is enough? And the same input could be made for ocean glint studies and rotational light curve measurements. We always have the chance to update our science gap list. Please look at the astrobiology biosignature gap number 16. Will be, if you're on the ExoPag mailing list, you'll see our message calling for inputs, which will come out sometime in the fall. And I would suggest that we know the target list at least to 0th order for HWO. If it gets bigger, telescope gets bigger, then the target list will get bigger. And there is such a target list that could be made for the ELTs also if you're doing reflected light imaging. And so the specific stars that are going to be accessible are to 0th order known today. And so if you're doing modeling of what a reflective spectrum would look like, I encourage you to pick one of the stars from the list and use that as the input to your models. Okay, that's it.
All right, questions for Carl?
Thanks, Carl. And this is also trying to reflect Bill Welsh's question in the chat. He asked, is stellar variability, flares, pulsations, oscillations, star spots, etc., included in the target star list? High variability may be confounding. So I look at the target star list, and are you going to be able, or somebody, or is there an initiative to provide more information? And are they going to look at test data to try to provide that, or what's the status of that?
So the target star list is now under the purview of the HWO Project Science Office. We sort of kicked it off before there was an HWO project by producing this document. And so now there is a broader list for which ours is like tier number 1, and there's additional targets that are considered. So in part the question goes to them. Variability we did not consider one way or another. It was not a scientific choice. If you could make a scientific choice of targets, you would pick some young ones, some old ones. You pick some of this spectral type, you pick some of that spectral type. You pick high metallicity and low metallicity. And those would be nice to be able to do all of that. We only concentrated on what was accessible. So I think as the list gets defined, we should track all these subcategories that are either good or bad for an HWO target, and try to see what would it take to modify HWO to be able to include more of those targets. I'm personally interested in seeing enough targets that have a Jupiter in them, because then you can understand the role of a giant planet in helping or impeding volatile delivery to the inner solar system.
A question, Carl, could you explain to people again what the ExoPag is, what that stands for, and how can it be involved?
Right, okay, so the ExoPag is the program analysis group, one of 3 in astrophysics, where we get inputs from the community. We hold meetings twice a year. The next one's coming up on June 14th, right before the AAS summer meeting in Pasadena. And so this is the community group that goes off and does analyses. So we've got a great ExoPag report on how the inhomogeneities on the surface of a star will make it very difficult to get spectroscopy of transiting planets, because the stellar background noise causes you to not be able to see the fainter features in the transit spectrum. So that there was a report on that. We had a report from a committee on a need to do more radio velocity work, and that led to NASA deciding to do a ground-based RV spectrograph for the US community and funding that. So the ExoPag has, at its meetings, we usually have about 80 people or 100 people at the winter AAS meeting. And then we have the committee that is working on this. They've produced about 25 reports over the years. And so the ExoPag is sort of our partner. Make sure that we're hearing from the community on what NASA is doing right and what it's not doing right in supporting the exoplanet community.
Yeah, Carl, I just wanted to emphasize your point about the near-infrared coronography. There are technologies being studied now that could push that inner working angle in, as you know, but they're low in what's called technology readiness level. And so this is why it's really important that the community really advocate if it's needed on what the requirements in the near-infrared are, because the mission and NASA overall needs to understand how valuable, how important it is to invest in these lower ready technologies in order to push that much closer. So.
Yeah, the HWO project is about to issue a call for instrument studies, and I'm hoping there will be many responses along the lines you've suggested, Avi, that how can we push in the inner working angle so we can see further into the infrared for more targets without imposing greater stability requirements on the telescope, which is one of the trade-offs you have in coronography.
One more question I have to ask. Given that was this last talk, do you think HWO could resolve the difference between a red edge at 700 nanometers versus one at 740?
700 versus 740. Well, the spectroscopy resolution is going to be good enough to do that, although it's really a question for Vicky and her committee. And in terms of how many targets can you do that, and I think you're comfortably able to do that for 2/3. I would say for all the targets that were on our suggested list, you're able to do that. It's just going past 1 micron where you start to really lose stuff, unless we get the new aggressive inner working angle coronagraph that hasn't been mature at all.
Okay, let's thank Carl again and the rest of the speakers in this session.